All right, so thanks everybody for making the trek across. That was confusing with a different room, so we'll probably get some people wandering in late. But I'm going to go ahead and get started, I think, anyway, just to kind of make sure I get through all of this, because fMRI of pain is a pretty broad topic, and I was asked to present that. Usually I've done more kind of methods-focused talks in the summer series. So I'm going to do my best to summarize what I think are some of the exciting issues in pain, but I'll say at the outset, I am not going into everything. So there's a lot to be discussed. Maybe next year, Dr. Bushnell could come. I'm not really going to be talking much about chronic pain at all. I'll say that at the outset. But I'm going to talk about why I think pain is compelling to study, and especially for cognitive neuroscience and affective neuroscience. So first of all, I think probably everybody here right now has heard a lot about pain recently because of the opioid crisis. So in 2011, it was estimated that 116 million American adults are affected by chronic pain. The estimated cost of medical treatment and lost work due to pain is $635 billion a year then. And then right now, already within the past 2 years, I had to update the statistic. Right now, the most recent estimate is that 115 American adults are dying from opioid overdose every day. So we really, NIH is focusing on pain in the context of the HEAL initiative, which is the new initiative helping to end abuse long term. And this is a slide from Nora Volkow's talk, which really puts pain management at the top of this pyramid of approaches that NIH is using to try to address this crisis. And so more broadly, in terms of why I think we should care about pain, we have two kind of aspects. So first, there's the clinical significance of pain, which goes into what I just talked about, but also if you're interested in how cognitive factors affect clinical outcomes, placebo effects, which are a great example of how the mind can affect the body, are largest in pain. I already mentioned about the statistics in the U.S., but also the opioid epidemic emphasizes how we actually need to work on more non-pharmacological kinds of treatments that affect pain. And we can use those either in conjunction with active pharmacological treatments or instead and help people taper off opioids. Some people here might be from NIMH. We've been collaborating with Danny Pine to use pain stimuli in anxious individuals. Pain is highly comorbid with anxiety and depression, and so it can be useful to look at these factors together. Now, also, as a basic neuroscience scientist, pain is useful as an experimental model. So pain is defined as a subjective experience. I'll go into some of the definitions and theories behind that. It's got both sensory and emotional components, so you can describe where the pain is or what kind of pain you're feeling, but pain's also unpleasant and you have limits to what you'll tolerate, and so that's kind of the affective component. The pathways that process pain are highly conserved across species, so we can use rodent models to develop treatments and test if they work in humans, and vice versa, identify pain disorders in humans and develop rodent models, and that's something we've been trying to do a lot in NCCIH. And then what I think is really cool, and this will come out a lot, is basically, unlike a lot of other areas of affect, we can objectively manipulate the noxious stimulus. So if you're interested in emotional processing, you might use something like IAPS or emotional images. Those are very hard to objectively quantify, but with pain, you can actually know the temperature that you're applying and then measure how a person perceives it. And so I think from an experimental standpoint, that makes it really easy to kind of know the objective link and then test how psychological factors might modulate that, and then where in the nervous system those effects are mediated. And finally, we have coordinated responses across the levels of the nervous system. So you can look at the brain response to pain, the autonomic response to pain, and the subjective response to pain. And sometimes we see that these things are all tightly linked, and sometimes we see interesting dissociations. So these are all sort of things that I find exciting to think about in the context of pain. Today I'll be talking about some background and definitions and sort of historical, philosophical issues about pain. Then I'll be talking about fMRI for pain. I'll give a kind of historical overview and then focus specifically on brain mechanisms of pain perception and psychological and pharmacological modulation of pain. And then finally, in the last section of my talk, I'm going to be talking about some of the controversies. I can barely even get into them. Pain has a lot of kind of exciting issues that people debate a lot. And so I'll give you guys a hint of that and kind of where you should go if you're interested in learning more. So pain is defined by the International Association for the Study of Pain as an unpleasant sensory and emotional experience associated with actual or potential tissue damage or described in terms of such damage. So you don't actually need the damage in order to experience pain. That's right there in the definition. And it's an actual, subjective experience. Whereas nociception is the actual encoding of the noxious stimulus. So there's a distinction between the subjective experience and the response to the noxious input. And that's pain versus nociception. Now, there's 3 different types of pain. So transient pain is what we're going to be talking about most today. This is defined, I like these definitions from Lozer and Melzak, as the activation of nociceptive transducers in the skin in the absence of any tissue damage. So this is kind of ubiquitous. I might bang my hand on the podium for a second. It might register as pain, but then I can move on without really having to go to a doctor. And so this is the easiest thing that to study in the lab with those kinds of experimental approaches. So most of the studies I'll be talking about use this device called a thermode. So it's basically a heat plate that straps onto your arm with Velcro or somewhere on your body and heats up to different known temperatures. And so you can apply a stimulus for 5 seconds, it goes back down to baseline, and you can measure how the brain responds. We can also use electric shock, pressure, so devices that create a certain amount of pressure, and laser stimuli. There's also capsaicin, which we'll talk about a little bit, which is the extract from the chili pepper, which can, as a topical treatment, can kind of elicit a burning pain, and then cold water bath. So these are a lot of different types of modalities that we can experimentally manipulate and then measure the pain and the brain responses elicited by those stimuli. Now, acute pain is kind of a continuum. So this is defined as substantial injury of body tissue and activation of nociceptive transducers at the site of local tissue. The pain is going to end before the healing occurs. So if you cut your leg, right, so you might experience pain for a while before the wound actually heals. And in this case, there might be a need for medical treatment to reduce your pain or expedite your healing, but eventually the body will recover. And this can be because of trauma. This is the kind of pain you would have in surgery. So if you're going to your dentist, you might have acute pain from getting wisdom teeth removed. So we're starting collaboration with the dental clinic here. Or for different kinds of diseases, they can cause this kind of pain. And finally, we have chronic pain. And that's when the intensity of pain is out of proportion to the original injury or tissue damage. It can be triggered by an injury or disease, but it can be perpetuated by factors other than the actual cause of the pain. So things like low back pain, diabetic neuropathy, fibromyalgia, or phantom limb pain. Obviously, especially in phantom limb pain, you can see there's no longer input that's causing that pain, yet it's continuing to go on. And that's kind of thought to be driven by the brain. So these are the 3 different kinds of categories, and I'm going to be focusing primarily on the transient pain because that's what we've studied for the most part in my lab, and also the field of fMRI is much kind of clearer on studying this kind of transient pain, whereas the question of studying acute pain is done very differently in a lot of different labs. I'll sometimes mention it, but I couldn't fit that into the talk as well. So when we're interested in studying pain, since pain is subjective, we of course have to make decisions about how to measure it. So this is the kind of classic combined visual analog and rating scale that has verbal descriptors going from no pain up to, in this case, unimaginable or unspeakable. Sometimes people use the maximum as most pain imaginable or most pain you could tolerate. So people use different anchors for different types of pain measurement. And there's a huge amount of work in psychophysics looking at the psychometric properties of these different kind of scales. So one of the issues right here is that somebody who has had, let's say, had a child or gone through different kinds of trauma in their life might have a very different idea of what unimaginable pain is than somebody else who hasn't gone through severe pain. So these types of scales can differ in terms of how different individuals use them. So there are scales that have no verbal descriptors and just use the visual analog, like the lines and the mark. Other scales that do the opposite. And so there's a whole sub area of pain simply dedicated to how to measure pain. We can also divide pain measurement into asking people to rate the intensity of the pain and the unpleasantness of the pain. And so these are also, we'll talk later about how these are thought to be segregated in the brain. And so one of the most recent pain scales I came across was the Neymar pain scale. So there's a lot of different kinds of pain scales out there, and we all have to make decisions about what we feel comfortable using. So going back, there's centuries of theories about what pain is and how pain comes into your consciousness. So Descartes has kind of this is one of those famous pictures in neuroscience, and it's kind of one of the foundational ideas for how pain reaches your brain and how you respond to it. So he believed that basically when the foot comes into contact with a painful stimulus, this kind of triggers a response that travels up this tube, and then it hits a gate right there at the pineal gland that releases animal spirits that cause you to react and move your body away from the source of heat, turn your body to see what the heat was, and respond to it. So again, you have this distinction between the nociception, which is the impulse traveling in, and pain, which is when it actually reaches the brain. And going forward to how this actually, more current theories of how this kind of transduction happens, there's about 4 different theories that pretty much don't even need to get to the brain to decide how pain is processed. So specificity theory suggests that there's different types of receptors that respond to noxious or innocuous stimuli, and that therefore you have what are called label lines that translate all the way up to cortex, and that lets you know if something was burning or stinging or pressure or aching pain. And those are processed through different types of receptors. A second idea was that it's actually the coding of the input that tells you what kind of stimulus it is. And so in this case, you're going to have the same neuron respond to both noxious and innocuous stimuli, but it's going to respond more when there's a noxious input. Then there is pattern theory, which suggests that the combination of representations across different cells is how you kind of can discriminate different pains. And this sort of, I think in the course you guys have already gotten a lot of exposure to MVPA at this point. So this kind of has the same idea that through the combination of different responses, you can actually classify what the stimulus was. And finally, the kind of account that was most compelling for about 50 years is the gate control theory, which suggests that at the spinal cord, there's kind of a gate or descending input that decides whether or not a stimulus reaches cortex. And the reason for having that is because it was known that sometimes the same stimulus is perceived as painful and other times it's not. And so this is the idea that within the spinal cord there are interneurons that can prevent something from reaching cortex, and also that your brain plays an active role in descending modulation and preventing certain stimuli from reaching awareness. Now, to show how these kinds of same theories actually account for brain responses, this is kind of what the specificity theory proposes. So the idea is that all the way up into cortex, you have specialized regions that respond to different types of inputs with specialized receptors at the periphery. The gate control suggests that it's really about this kind of inhibition and filtering that happens at the spinal cord. And then the theory that kind of accounts for most of what we think about when we're looking now at cortical responses to pain is the pain neuromatrix theory. And this suggests that within the brain responds as a network, and that network can code the sensory discriminative aspects of pain, so that intensity perception or what type of pain, the motivational and affective components, the unpleasantness of pain, and the cognitive or evaluative components like your attention to the pain, whether your attention is drawn. And so when we talk about different studies, this is kind of the main framework that people use for understanding how the brain processes pain now. And Melzack also said that as a function of these kinds of combined networks, then you decide in parallel processes how you perceive it, how you respond to it, and how to regulate your body's response to the pain. So that's all like nice and theoretical. What does the data say about the brain's role in pain? And again, this is all before brain imaging. So basically, lesion work, originally Head and Holtz said they looked at patients with parietal lesions. They saw that pain was not affected. They decided that the brain is not necessary for pain. But others showed that you can reduce pain when you have cingulotomy or lesions to somatosensory cortex, and those can also create pain, suggesting that the brain does matter for pain. Brain stimulation studies were thought to, again, indicate that the brain's not necessary for pain. So Penfield stimulated somatosensory cortex with 800 different stimulations. Only 11 of those elicited ratings of pain at all. And there's a nice quote. I don't have the presenter view now, but it's basically like, therefore, we've concluded that the brain does not contribute to pain in any way, and something along those lines. However, other studies have shown that stimulation can elicit pain, and in implants that deliver stimulation are also involved in relieving pain. And finally, there's some beautiful work that's been done in primates doing electrophysiology with tests where monkeys can kind of set their own threshold for what's too painful for them. And these studies do show that there are nociceptive neurons in the thalamus, S1, and some secondary somatosensory cortex. However, it's really hard to ask a monkey how painful that was or whether or not the intense stimulus was painful. So we've got brain imaging now, and so we can actually kind of try to tease apart these networks and see how they're implemented and what role does the brain play in constructing pain. So the first studies done looking at the human brain pain were using this thermode heat pain model. So again, it's heated up to different temperatures, up to about 120 degrees Fahrenheit or 50 or so degrees Celsius. And so the first study to show pain in the human brain using imaging was done by Catherine Bushnell. There's people from her lab here. A number of these studies were by Catherine, comparing PET cerebral blood flow responses to painful heat versus warmth. And so there they confirmed that indeed primary and secondary somatosensory cortex and anterior cingulate all do respond more when a stimulus is painful than when it isn't. This was also seen by Anthony Jones the same year in anterior cingulate as well as the thalamus and subthalamic nuclei or lentiform nucleus. The first kind of evidence that your psychological response to pain plays a role in how your brain responds is from Bud Craig, also in Catherine's lab group, looking at the thermal grill illusion. So this is basically when you repeatedly go between 40 degree stimulus, which is not painful, and a 20 degree stimulus, which is cool but not painful. When those are combined back and forth, that creates pain. And so the stimulus coming in is exactly the same, but when you have that illusion, it's painful. And so what they found is that the anterior cingulate responds no matter what, or responds when you perceive a stimulus as painful, whereas the insula is responding to all of the stimuli, right? So the anterior cingulate seems to be specific for pain here. And finally, they went further to actually look at the cingulate and somatosensory cortex response as a function of intensity versus affect. So they used hypnosis to help people experience the pain but find it less unpleasant. And they found that that reduced responses in the anterior cingulate. So these studies all showed that indeed, the anterior cingulate responds when something is more painful, as does primary and secondary somatosensory cortex, and begins to talk about how psychological factors can actually modulate pain and brain responses to pain. And the reason this is important, just to back up a second, is because pain is so clinically relevant, but we don't have a biological measure for it. So it's the number one reason people come to the doctor, but then there isn't actually a test to see if somebody's pain is real. So now we're seeing that somebody saying they're feeling pain is also accompanied by real biological changes in the brain. And we'll come back to that question. So after these PET studies, people began using fMRI. So these are the real early studies. They didn't have color images then. So you can see here, this is with electrical nerve stimulation. So they didn't have an MRI compatible thermode back in those days either. And so what you can see here is they get contralateral S1 activation as well as anterior cingulate activation in response to painful electric nerve stimulation applied to the arm. They don't see it when the stimulus is not painful, and it corresponds with the patient's time course of pain reports. So that's a big advantage, obviously, in the switch from PET to fMRI. They could start looking at the dynamics of responses. Then people have done esophageal distension. So this is basically swallow a balloon that opens up, gets at visceral pain. And so again, here you see cingulate and insula, as well as some kind of operculum activation. And then here is a study using capsaicin. And this is kind of like you put the capsaicin on, that gives you a little bit of a sensation. And then when you apply heat to it, it exacerbates it. And so here again, they see S1 every time that they apply the capsaicin. And so in 2000, there were enough studies of pain in the brain to do the first meta-analysis of pain responses in brain imaging. And so what you can see is that we do see pretty consistent activation of bilateral insula, anterior cingulate, some somatosensory cortex, but that was a little bit less consistent across studies, but also potentially because they were stimulating different locations. And secondary somatosensory cortex was also, that was the region that responded most in all studies. And even going back to this original meta-analysis, what we see, and this will be in the controversies section, is that the cingulate shows overlapping representations between pain, here in red, and attention in green, unpleasantness in light blue, and then stress or mood causing deactivations in these regions in dark blue. So you have this question of, is what we're seeing when we see activation response to pain really just about the fact that the stimulus is unpleasant, or is it something specific about the nociceptive response in that region? And that's really something that has kind of stayed one of the themes of this kind of research for almost 2 decades now. Now, in this meta-analysis, they also show there's overlap relationships with looking at pain in chronic pain patients here, and also that we see reductions in some of these regions when we look at brain imaging during analgesia, so when somebody's getting an opiate or some kind of treatment for pain. So it's really focusing on the cingulate, and we see pretty robust activation, but of course the cingulate is also involved in many other types of functions. So this is a more recent meta-analysis, and I'm just going to flash it up quickly because you can see pretty much the same systems are activated. Now we're better at seeing subcortical responses, so we see the striatum's highly involved, the midbrain, and the cerebellum. But for the most part, we kind of have come to a pretty consistent understanding of what to expect when we're measuring pain, and this is the pain matrix or pain neuromatrix. So this is a mini meta-analysis across 5 studies that all applied high and low stimulation. And so this is a contrast between them. And so just to kind of, give you the summary on that, we've got brain regions that are thought to be involved in pain's sensory components. So that's lateral thalamus, primary and secondary somatosensory cortex, and the posterior insula. And brain regions thought to be involved in the affective components or the unpleasantness, more medial thalamus, anterior cingulate, and anterior insula. And finally, we also see reliable involvement of the PRG cerebellum and striatum, which might be involved in that kind of motivational motor prepared response to escape the pain. Now, some colleagues and I recently wrote a review for non-imagers and new trainees to talk about kind of what considerations are unique to pain imaging when it comes to fMRI. So either for people who've done fMRI before and never thought about pain, done pain work but don't really know about fMRI, we kind of tried to highlight some of the unique considerations. So I thought they'd be interesting to mention to you guys. So first of all, we can't have any metal in the scanner. And that really actually rules out some pain patients because people might have implants to, if they had hip surgery or something. Or some people are really interested in studying phantom limb pain, but a lot of the amputees have devices that they need. So that kind of creates some challenges. And those of us who are studying pain even right now, our heat stimulus that we use, the Thermode, is like a really finicky piece of equipment, and there's only one company that makes an FDA-approved MR-compatible pain device. And so we all have to use it, and it breaks all the time. So basically, doing pain research in the scanner can be difficult. Our patients can't move in the scanner, but we're applying a stimulus that should be painful and should make people want to move. So in the end, we may not actually ever be able to hit that acute pain level in the MRI scanner because we have to minimize motion. So that kind of, again, is sort of a question about the ecological validity of this. One of the things that actually made us write this paper is because my friend, who's the first author, whose name I won't mention, didn't know that he should report how many people dropped out of his study because it was too painful. And I'm like, but other people are going to do the same experiment, so we actually need to know what proportion of people can tolerate the pain you're applying and what proportion can't. So we have to think about that in our papers. Who actually enrolls in pain studies? It turns out that men who perceive themselves as extra manly are more likely to decide to volunteer for pain studies, and also people who are higher in sensation seeking. So we hope our healthy volunteers are representative, but there is, a chance that we have some unique people. And finally, one other question is specificity to pain, and we're going to get to this at the very end of my talk. You guys can ask questions or anything throughout, but I know sometimes in these series people don't really speak up very often. So that's kind of a broad overview. Are there any questions on that? Okay. So the next thing I decided to talk about was actually brain mechanisms involved in pain perception. So the studies we talked about so far have all just compared a more intense noxious stimulus, where it's known to be higher, with a less intense noxious stimulus, or even pain versus rest. But those original studies didn't actually look at the subjective pain experience itself. And so the next kind of big foray in fMRI was to actually look at the subjective pain experience. So the first study that I think is really cool, and might be one of the first studies to do this, was by Vanya Akarian, looking at differentiating cortical areas related to pain perception from brain regions involved in stimulus identification. So he basically applied 5 seconds, or no, I guess 35 seconds of painful stimulation. And this is the average across a group of 8 subjects of the pain experience. So like the heat stimulus is on, it gradually ramps up. 50 in their scale is the most pain imaginable. So 30 is like pretty high pain. And so basically like the experience goes up, and then when the stimulus comes off, they go back down to 0 pain. So this is the time course of the subjective pain experience. Ignore this correlation with pain for now. And here, it's a chart. And so what they did is basically collect, how many slices? Like 10 or so coronal slices from anterior insula to like posterior insula. So that includes insula, anterior cingulate, thalamus, they don't really talk about thalamus, and S2. So you're kind of getting this gradation, and we're not going to see any pictures of brains in this study. So basically, what we see here is the time course of that pain convolved with an HRF. He both used the standard HRF and also verified that that HRF did account for activity in the ROIs. Then so that's the solid line. In this kind of dashed line, you see the stimulus time course. So this is like when the heat was applied, this line here, convolved with the HRF. So you can see there's a difference in the time course of those two. And then in this dashed line, the jagged one, is the average time course of the pain ROIs from this kind of segment of the brain. And so what he found is that the ROIs correlated each of the ROIs, there were like 12 ROIs that he made bilaterally, correlated most strongly with the stimulus time course rather than the pain time course. But then when he looked at the correlation with the pain time course, he found that the correlation with pain was higher at more posterior regions. So this kind of highlights the dorsal posterior insula, which is a region that many studies have kind of triangulated on as being involved in the subjective pain perception. Some people think it's like the region. So this basically goes from anterior insula, which is responding to all the stimuli, like that one map I showed with the thermal gill illusion, to posterior insula, which is most related to subjective pain sensation. Then moving further, when people started doing event-related fMRI, Christian Buchel's group started looking at how a brain response could be related to pain intensity, stimulus intensity, or stimulus awareness. And so they did this with this combined pain scale where people rated it 0 if they didn't perceive anything, 1 if they perceived it as warm but not painful, and 2, 3, and 4 were graded pain sensations. So this is what the response would look like in a brain region if you weren't aware of the stimulus at all. This is what it would look like if you're just tracking stimulus intensity. And here's what it would look like if you're tracking intensity and it's specific to pain. And so what he found is that within the anterior cingulate, he found all those different kinds of patterns. So basically there were voxels that responded to pain intensity, voxels that responded simply when the subject was aware of the stimulus, and voxels that responded to the stimulus intensity over time. And so this kind of like shows you that this, all or none, is it just about the stimulus or just about the pain, is a little bit simplified. And we'll get kind of more into this. They ran another study, they published the same data in another paper talking about the rest of the brain. And so other regions kind of showed all of those patterns as well. So here we start seeing inverse responses in the ventromedial prefrontal cortex. We see that the dorsolateral prefrontal cortex is it responds when you're aware of pain, irrespective of the actual intensity, and other regions track the intensity of the stimulus or the intensity of the pain. Now, what we wanted to do is actually kind of use mediation analysis to look at the direct relationship within people between temperature and pain. So last year I talked just about mediation analysis, so if you want to know about the methods behind this, you can look at my video from last year. But we wanted to know which brain pathways actually respond to temperature and predict how much pain you report on a trial-by-trial basis. So basically, mediation tests if the relationship between some input variable and some output variable is reduced when you control for an intervening variable or a mediator. And if it's a significant reduction, that's called mediation. So what we did is single trial analysis where we can extract the response to every trial over time. We use slow event-related analyses to do this. And so that gives us for each individual, we've got the temperatures that were applied over time, the pain ratings for that individual, and then we've got the brain responses during heat for every voxel. And so what we're basically asking, well, I'll explain it in the results, but so what we do is we test every voxel. Does that voxel significantly mediate the effect of temperature on pain? Does it account for some of this correlation? And so this is what the correlations look like overall across subjects. So you can see something I'm not talking about in this presentation is individual differences in pain threshold or tolerance. So people can be highly variable in terms of what temperature they begin to feel pain. So this is on the low end, and what temperature is the max that they're willing to tolerate. So we're looking, but instead of that, we're looking dynamically within an individual. So here, let's say, this is the exact same stimulus, but sometimes this subject's calling it painful and sometimes it isn't. So what accounts for that relationship? So first, our mediation looks at which brain regions show differences as a function of temperature. And so what we see is pretty much the whole pain neuromatrix in yellow, as well as regions that are inversely related to temperature. Kind of default mode, I guess, is one way to think of it. And then we also have brain regions who predict pain controlling for temperature. So that's that variation within that one subject's orange scatter plot. So basically, regardless of the temperature, the higher the activity in these regions, the higher the pain. And again, you see that these regions are not just driven by temperature, they're also predicting subjective pain. And finally, we looked for brain regions that formally mediate temperature effects on pain. And so what we found, again, is that pretty much most of the pain network does formally mediate temperature effects on pain, particularly the insula, the striatum, and the anterior cingulate. But we do have some somatosensory activation as well. So that means the more temperature, the higher the temperature, the more activity in these regions, and controlling for temperature, the higher the activity, the stronger the pain response. And we saw we did a network-based analysis to segregate these regions into different networks based on connectivity. And what we found is that actually, look, it was interesting for me to put together this talk, but we found the same kinds of patterns that Buchel actually found when he looked at those individual regions. So some regions show much more linear effects, going from non-painful warmth to maximum pain tolerable, whereas others show responses that are specific to pain or that are kind of inversely related to pain. And the last thing about this study is that we also found gradations within the anterior cingulate and the insula that had portions that were related to temperature but not related to pain. So that's path A only. Portions that are related to pain but not related to temperature. So that might be something, well, I'm not going to speculate about cognitive process. And portions that showed mediation. And interestingly enough, we find that same anterior to posterior gradient that Apkarian found in his early study, covering these same regions. So I didn't really notice that, I don't think, when I was writing up the paper. But so here we are kind of using new methods that can computational sort of understand the dynamics within an individual that relate temperature and pain. And we're coming to the same conclusions that earlier work did, using just kind of the time course of when the stimulus was applied or when pain was perceived.
So how do you see on this plot the anterior to posterior gradient?
In this or this?
In the brain.
In the brain, just this is more posterior than this. So basically, in this plot, the correlation is how correlated is it with pain, and low values are more correlated with the time course of the stimulus. Mm-hmm. And so for us, the voxels in yellow show correlations with temperature across trials. So it's different. It's not the time course of the stimulus. It's instead the trials that you're getting a 42 versus trials that you're getting a 45. So the yellow regions show increased activation as a function of temperature, but don't show any additional relationship with pain, whereas the red regions don't respond at all to changes in temperature, but within a temperature predict variations in subjective pain. Does that make sense?
Yeah, that makes sense.
Okay.
I wonder if you know about Daniel Margulies'
yes.
from sensory to associative areas. Does this look like the same type of?
Oh, interesting. Okay, so let's talk about that at the end, maybe, because I think at the end we'll talk about multisensory questions, and that might be relevant. So I know the name, but I actually don't know these papers, so I'd be really curious what you think. So just to summarize this study, we're finding that pain is generated by a combination of independent networks, and mediation helped us identify regions that link the objective stimulus and subjective response. I'm going to have to kind of go quickly through some of the other stuff, but I definitely want to go through it. So I'm not going to rush crazy, but I'll just be mindful of the time now. So we actually then took the same data and we built a classifier. Let's see here. So we used, how many of you are familiar with Neurosynth? Okay, so Neurosynth is an automated meta-analysis that basically pulls terms and voxel tables from a whole bunch of different journals to come up with automated meta-analyses. And so basically, we used Neurosynth to create a pain mask, which essentially is the same regions we've been talking about through the talk, and then used Lasso with principal components analysis regression to generate this classifier, which is called the neurologic pain signature. So this is basically an unthresholded map, but we usually look at it like this, the regions that were most predictive of pain. And so we trained it on the data that I just showed you to generate this pain map that predicts basically whether or not something is painful and which of two stimuli are more painful on a trial-by-trial basis within people, but then did a cross-validation to make sure that it predicts other subjects in that same sample. We then found out, fortunately, that classifier was not only good at predicting other subjects in our study, it also could predict other studies that we'd run in the lab and also other studies run in other labs in other scanners of other types of conditions. So basically what happens in this is kind of, we've got this pattern of weights, and with any given contrast, you can just multiply your beta or your contrast value by the classifier to see whether or not something is more painful than another condition. So just to kind of show you, so, you know, in the original paper, we showed that we're highly accurate at predicting high versus low pain, pain versus romantic rejection, and we are not good at predicting somebody rejecting you versus a friend. And we'll talk about that at the end also. So it's highly specific to pain. When we looked, I just thought this was pretty interesting, so I was looking at the supplement, and so first of all, on a trial-by-trial basis, this is what the classifier predicts. Based on the classifier, the predicted temperature, and this is the temperature that actually was applied. So we're pretty good at predicting the actual stimulus that was applied, but then when you look at, just like Epkerian did, the difference between the time course of stimulation and the time course of pain, the classifier is much better at is much more correlated with the time course of pain report than it is related to the stimulus. So this is really a classifier for the experience of pain rather than just the response to nociception. And that is further shown here based on, this is another study that was run in Colorado instead of New York, but just basically multiplying the same classifier with those beta coefficients. And what you see is that, so on the y-axis is the signature response, and you see when a subject doesn't perceive the stimulus as painful, you see it's not very good at telling the difference. However, as soon as somebody starts perceiving it as painful, which is above 100, then it becomes really good at differentiating the response between stimuli. And we and Tor's lab, without me, have built on this in many ways. So they've got a vicarious pain signature, so when you see somebody else in pain, a stimulus intensity independent pain signature, so basically controlling for temperature, something that just predicts those variations in pain, the way I was talking about in the mediation analysis. We've got an approach that can basically take this classifier and kind of predict for individuals rather than just the group, and then integrating that classifier approach with multiple mediators is a new paper on bioRxiv. Now, others have also used this same kind of classifier approach in the context of chronic pain. So Epkerian's lab showed that connectivity between the medial prefrontal cortex and the nucleus accumbens can predict who's going to go from acute knee pain to chronic pain over the course of a year with high accuracy. And Marina Lopez Sola, also in Tor Rager's lab, developed two different classifiers, one that predicts fibromyalgia based on responses to painful pressure, and another that predicts fibromyalgia based on brain responses to multisensory input. So basically, this kind of approach kind of is getting more at what this neural matrix, what is the combination of networks and their responses that can help differentiate between pain and just the noxious input. Since that was going to be my only other place that I'm mentioning chronic pain, I just wanted to at least kind of acknowledge that meta-analyses do suggest that in some cases we see overlaps, such as in the insula and cingulate, between experimental and chronic pain. But in other cases, we see differences. So here, there's portions of the insula and posterior cingulate that are seen more in experimental pain, and there's other portions of posterior insula and prefrontal cortex that seem to be seen more in neuropathic pain. So other labs here have more expertise in this than me, so that's I'm going to continue. So just with the last part of my talk, I'm going to quickly go over how psychological factors modulate pain. And so here's a review from Catherine Bushnell showing the ascending pathways that we've been talking about so far in gray, and then how these can actually be modulated in the context of either emotion and the placebo effect, or more cognitively through attention. And so there's a lot of different studies manipulating each of these factors to try to understand the extent to which they actually modulate the brain's response to pain. Because again, these factors might affect the pain that you say you feel, but are they really affecting those brain regions that we think are involved in the primary kind of response to pain? And so the way that we do this is basically by one area that we've focused on a lot is expectations. And so what we can do is we can use cues that, like through classical conditioning, that predict different levels of pain. We can use placebos, which tell people to relieve, that their pain will be relieved, but there's no actual pharmacological substance. And then we can actually deliver analgesic treatments and stimulate pain to test how pain is modulated by active pharmacological agents. So basically, in a stimulus expectancy manipulation, people might be told what pain they'll get following cue and or undergo conditioning. And then we compare those cues with a given stimulus and measure how much pain that elicits. And similarly, in a placebo study or treatment expectancy manipulation, we can tell people that some fake substance is a really potent pain reliever. We'll apply that and reduce the noxious stimulus relative to the exact same skin cream that somebody thinks is actually a treatment. Nobody in this room is allowed to volunteer for any of our studies. And then we can, during a test phase, apply both stimuli and measure whether pain and brain responses to pain are reduced on the site that was treated with a placebo. And so looking at how that NPS signature responds to these, so what we see, first this is a meta-analysis that we conducted across all types of studies of expectancy. And so we see reliable reductions in the insula, thalamus, and dorsal and anterior cingulate during pain. So those are those regions we talked about as involved in the affective components. And we see increases in the ventromedial prefrontal cortex, the dorsolateral prefrontal cortex, and the striatum. And so these regions are thought to be involved in both value learning and kind of cognitive control. And a lot of people in the placebo field have tried to tease apart like which of those processes are kind of underlying these responses. Now, we've since gone back with the NPS and tested how that responds as a function of the cues. And so what we see is that, so in this case, when the same stimulus is preceded by a high pain cue relative to a low pain cue, we see strong differences in the NPS response. So it's not activated at all to the same stimulus when it's preceded by a low pain cue. But when we look at the placebo analgesia, a meta-analysis of that suggests that even though people report significant reductions in pain, we see no reductions on the NPS. So this suggests that placebo pain reductions are being constructed not via the NPS, through some other type of pathway to drive pain in that experience, where these types of cues are affecting the NPS. Does that make sense? People following that? Okay. I'm going to skip over this study and just get to, well, I'll just say that people are now talking about also being able to use classifiers to test if drugs are working. So basically, this is from my Reed Tracy's lab. What they did is they looked at brain responses to pain and responses to drug relative to placebo and developed a classifier that is predicting how much a brain region differentiates between drug and placebo. And then by testing multiple different drug trials, they can test if that classifier can really measure whether that drug is relieving pain. So basically, kind of which drugs are reliably relieving pain across multiple sessions is the idea that you could use a brain-based classifier for that. So this kind of gives you some sense, and I skipped over the study where we combined pharmacological and psychological factors just in the interest of time. But with the last 3 minutes or so, I just want to highlight the topics that I mentioned slightly but don't have time to go into, so that if you guys are curious about pain, you have some fun reading. So basically, one big question is when we experience rejection or sad feelings, are those really painful? So the idea is kind of, if you've got a heartache, or we use a lot of terms that are related to pain to talk about our emotional experiences. And so many psychologists and cognitive neuroscientists have thought that these are interchangeable, basically, that what's happening is when you experience rejection, you are experiencing social pain, and that your brain responds in the same way. So here's a meta-analysis of brain regions when you experience pain yourself in green, and when you see another person experience pain. And the regions in red are the intersection of those. So you can see that there are some regions in the anterior insula, the anterior cingulate, and the ventral anterior insula, basically cingulate and anterior insula, that respond both when you are in pain and when you see somebody else in pain. And this is, I don't remember how many studies went into this meta-analysis. So Naomi Eisenberger basically had the first fMRI study asking whether rejection is painful. So she developed a game called Cyberball, where these little characters are tossing you the ball, and you toss it back to them, and you choose who to toss it to. And then all of a sudden, player 1 and player 3 start passing it back and forth, and they stop passing it to you. And in the original version, there's a hand at the bottom of the screen, and it's just watching the ball go back and forth and waiting for them to pass it to you, and people don't get it. And so basically, the more distress somebody reported as a function of that social exclusion, the higher the anterior cingulate activity. And so what Eisenberger, and this was kind of replicated in other studies that showed that basically social distress was also the lower your pain threshold, the more unpleasant pain, the earlier you experience pain as unpleasant, the more distress you reported on that task. And studies have administered Advil or Tylenol and showed reductions in the exclusion, the feeling of exclusion. So really implying that these are the same process, right? That exclusion feeling and the feeling of pain. So here are some studies reacting to those original papers. So basically, anterior cingulate responds differently to expectancy violation. It's not about rejection, it's about expectancy violation. This is really about conflict monitoring. Then using machine learning to show that you can have separate representations for physical pain and social rejection, even within the same portions of insula and cingulate. So simply showing cingulate and insula is not sufficient to say it's the same representation to your brain. And it comes back to that notion of patterns. And the idea is that, yes, there might be something going on, but it might not be the same to your brain as being in pain. This led to something, if you're on Twitter, you can search hashtag CingulateGate. So basically, Naomi and Matt Lieberman did a meta-analysis using neurosynth to say that the dorsal anterior cingulate is selective for pain. And I'm not going to read all the titles from the blogs that followed, but basically this was a long back and forth about, first of all, whether they used the meta-analyses properly. Secondly, is it really selective? There's a lot of reading to do. So one big question is basically, can you look at the brain and do reverse inference and assume that you're seeing pain? This kind of brings us to the broader question, which is, what is the specificity of these brain responses? So I'm pretty convinced, relative to the people, when we were looking at the lesion data and the electrical stimulation, yeah, the brain is involved in pain in humans. But is that what we're seeing in these studies? And if this is what we're seeing, can we actually look at somebody's brain and infer that they're in pain? So there's a lot of papers now about the ethics of doing kind of using brain-based biomarkers for pain. The idea is these biomarkers might help us make discoveries about research or kind of test within a given study, are you really eliciting the process that you think you are, but having a biomarker can be used in unethical ways if people aren't thoughtful. So for instance, if this patient says he's feeling pain, but his brain doesn't light up when they put him in the scanner, does he really deserve treatment? And this is a really important question. Well, we're almost out of time, so it's okay, because we have health disparities in pain. So basically, pain is defined by the patient's subjective experience. So even if we don't see these same brain systems activated, that might just mean that the pain is constructed by a different mechanism for that person, but the person's pain still deserves to be treated. On the other hand, there are people, such as minimally conscious individuals, who can't say what pain they're feeling. So this is a really interesting kind of ethical paper to think about what happens. So they actually applied painful stimuli to these individuals who obviously couldn't consent, but their family members did. And you see activation that looks pretty similar between the person who can't say anything and the healthy volunteers. And so having a brain-based classifier could be really useful for deciding whether or not to treat these patients. Let me close this. Finally, I've been talking about the anterior cingulate and the anterior insula, and most other people who don't study pain would think of this as the salience network. And so, this is a meta-analysis from Neurosynth, and even with reverse inference, the idea that if I see activation here, it's most likely to be pain, we're seeing the anterior insula and the cingulate. So can we even measure pain? In all these cases, whenever you give something more painful, it is also more salient. So maybe we're just seeing really, really salient stimuli. And the last thing I'll present is what I think is one of the coolest studies of this, which is a study by Moreau in Ianetti's lab that basically shows, first of all, how much overlap there are. All these voxels, so they delivered painful, innocuous, auditory, and visual stimuli, and all the voxels in yellow were activated by all the different stimuli. So the only stimuli that were selective for somatosensory painful stimuli are like these little voxels in red here. And what they showed is that even that they could decode basically. So they looked at the more salient stimulus in each of the modalities, and even in primary sensory cortex for each of those different modalities. So you go to V1 or primary auditory cortex or primary somatosensory cortex. Here's secondary somatosensory cortex. And you see that no matter what the modality is, so red is nociceptive, blue is auditory, everything except visual here, you could see differences as a function of salience in that region. So this really kind of brings the question of what are we looking at if our primary sensory cortices are that selective for other types of modalities. So I'm going to stop there. So what I've talked about today is primarily transient and acute pain. I do believe it is in the brain. That's why we study this using imaging. And that I've talked about how it can be dynamically modulated by psychological factors. I think this all kind of supports the notion of the pain neuromatrix, and these kinds of new network and pattern-based analyses are getting us closer to be able to segregate different portions of this complex experience. But there's so much left to understand. So first of all, I didn't touch on chronic pain, which is kind of the most important thing to be thinking about right now. We didn't talk about how we can leverage animal models. We're currently doing tests in our lab to actually try to get at specificity by applying different types of stimuli. So in collaboration with people at NIMH and John, we're comparing pain with taste just to look at that question of general salience. And then integrating fMRI along with other types of multimodal measures is really important. So things that have better time resolution, pharmacological effects, and actually considering individual differences and things that moderate the effects I showed you today. So there's a lot. I couldn't squeeze it all in. Thanks for your patience, and we have time for questions. We started a little late. Yeah?
I know that the presentation didn't really touch on chronic pain that much, but I'm curious in the way you're looking at a crisis, and there's this whole question of pain management. Do you see this data and this work in fMRI affecting the clinical application or the clinical addressing of under or over treatment of pain?
Okay, so that's a good question. So the question is, I have to repeat it here. So do I see a way that fMRI can be useful in quantifying under or over treatment of pain? Yeah.
Or like treating, because there's all sorts of like social issues, right? There's like historically women are undertreated for pain,
right.
And then when patients come in with chronic pain conditions and then they have an addition of acute pain, they're much more likely to not get treated for that acute pain.
So that is why I kind of kept it simple for this talk. So basically the complicated moderating factors, so for instance, like even the placebo effect, so I'm just kind of thinking of Bushnell, the placebo effect seems to work differently in patients with chronic pain, right? So basically, like even something that we've done a lot of studies in healthy volunteers might be totally different once you actually get to chronic pain and how that changes brain systems. So I kind of think that's one case. That's why I wouldn't extrapolate from what we see in healthy volunteers to assume it applies to chronic pain. But with the question of over and under treatment, I think that like some of these classifier type approaches are being validated in patients and seem to be working in patients. I think that studies of kind of addiction and substance use in general do target some of the same systems that we see modulating pain similarly, like in terms of cue-based craving and stuff like that. So I think that there is a lot of, there's a strong advantage to using brain imaging to try to understand different types of patients' pain, but I just couldn't do that justice in the context of trying to get this stuff across. And I think with health disparities, we're trying to understand that. So a lot of pain studies just study white men, like a lot of the literature. And so understanding kind of where there are sex differences or differences based on race, that's important too. And we do see differences in pain tolerance, both as a function of sex and race. So incorporating that into our studies is also really important.